Chapter 3. Continuity and leaps. Organisms are required to constantly adjust their energy management mechanisms to remain active, obtain energy security, and carry out life activities in an energy environment full of uncertainties. Such mechanisms encompass energy balance, energy intake, energy lock-in, as well as information reception and response, serving as the necessary components to withstand natural selection. However, the two-sided nature of energy use and the inherent defects of macromolecular structure make overcoming uncertainty and resisting the pressure of increasing entropy impossible for organisms. The question of life and death constantly looms over every organism, like the sword of Dartmouth. Thankfully, our earliest ancestors did not attempt the impossible. Instead, they wisely adopted the continuity and leap strategy to solve the issue. Currently, this strategy has proven to be successful and has been passed on to countless individual organisms. These two basic life strategies have evolved into various sub-strategies with different strengths over generations. Lifespan. All organisms possess a lifespan, which represents one of the fundamental strategies of life. This mission is given to every living creature from birth, and without it, life activities cannot be possible. Ancient ancestors adopted this strategy to uphold individual activity, achieve intergenerational alternation, and ensure the continuation of their life. The finite lifespan of an organism may seem like fate. Despite any regimen efforts made, the maximum survival time of an organism is still subject to constraints from a mechanism in the species' lifespan strategy. It is possible that an innate fundamental energy allocation mechanism exists in an organism. Starting from the formation of a fertilized egg, all activities of the organism are governed by this mechanism as a script. The primitive energy is likely to be the only factor in the mechanism for carrying out tasks related to activity and longevity. The stability of the primitive energy is critical, and every species must maintain its certainty to avoid any uncertainty factors. Failure to maintain certainty could lead to the misallocation of energy and the disintegration of the organism. Considering this circumstance, the common ancestor of the creature established the primitive energy as non-renewable and non-recyclable. This action prevented the vicious cycle of energy to the maximum extent possible. The amount of the primitive energy present can be determined at the start of fertilization or division. Members of the same species may have approximately equal amounts of primitive energy because they share the same initial energy value. Yet, the level of primitive energy in an individual may be linked to the environment in which the fertilized egg was created. The greater the stability of the environment during fertilization or division, the closer the primitive energy is to the baseline. Alternatively, the more significant the deviation is. It is challenging to experimentally verify the primitive energy hypothesis within the current scientific framework. Nevertheless, comprehending life's essence is tied to the presence or absence of primitive energy, which will be elucidated systematically in Chapter 5. The average lifespan of both animals and plants is often determined by factors such as the habitat, accidents, disease mechanisms, and several other variables. Determining the average lifespan of a population is challenging, and it's even more arduous to observe the average lifespan of a species. Additionally, studying the average lifespan of a species doesn't hold much value. Maximum lifespan is the longest lifespan achieved by an organism within a population, rather than a more generalized concept. The maximum lifespan of an organism is likely related to the primitive energy of its species, natural life mechanisms, individual growth pathways, and environmental changes. Most scientists in the research community prefer to focus on studying maximum lifespan. The lifespan of plants is still a mystery. There is a saying that the iron tree blooms only once in a lifetime. Iron trees have a relatively short lifespan. Mushroom plants live the shortest among our standard plants, followed by rice, corn, barley, and other types of grass. Trees, on the other hand, have a significantly longer lifespan than other plants. Tall trees such as pine, cypress, fir, etc. live considerably longer than fruit trees, oil tea trees, and other cash crops. Botanists typically classify the lifespan of plants into annuals, biennials, and perennials. The given information indicates that apples, grapes, and pears have a lifespan of 100 to 400 years, while elms, birches, and camphors can live for 500 to 800 years. Pines, ginkgos, and spruces have the longest lifespan of 1,500 to 4,000 years. Additionally, Welwitschia mirabilis, a desert plant, has 8-meter-long leaves that can survive up to 2,000 years due to its unique adaptation of just 2 leaves that grow indefinitely without fading away. Although there is human speculation about the lifespan of plants, there are no exact figures from all sources to determine animal lifespans. Life expectancy varies in harsh wild conditions. For instance, rats on average live for 1 to 2 years, whereas cats can reach up to 10 years and lions can survive for up to 20 years. Prehistoric humans, however, had a life expectancy of nearly 20 years only. In contrast, elephants have a life expectancy of 50 to 60 years, and some whales can live for over 100 years. It's apparent that insects and organisms that are of small size generally have shorter life expectancies than larger mammals, and microorganisms have even shorter life expectancies than insects. A cell's capacity to divide is typically equivalent to its cellular lifespan. In a given species, a cell's potential for division is typically limited to a certain range, determined by the primitive energy allocation mechanisms. Plant and animal cells have differing lifespans, typically with plant cells having longer lifespans than animal cells. When compared to animal cells, plant cells are less bioactive, more malleable, and thus slower in their division process. In contrast, animals exhibit higher bioactivity, higher metabolism, more frequent cell division, and a faster loss of primitive energy. Apart from the primitive energy being the foundational determinant, the maximum lifespan of a species depends on factors like metabolic rate, energy lock-in strategy, and environmental variability. Suppose a taxonomic group of organisms has the same primitive energy levels and natural lifespan, without predation, severe disease, and accidents. In that case, if one of the species follows any of the below-mentioned strategies, their lifespan gets affected. A narrower distribution of energy loci, more aggressive energy intake, more frequent defense and interception in energy locking, and higher repair and renewal capacity. By deploying these strategies, bioactivity increases, and the primitive energy consumption rate increases inevitably. These strategies certainly come at the cost of a shortened lifespan. Scientists from Kyoto University in Japan and the Australian National University analyzed the photosynthetic mechanisms of hundreds of plant leaves from all over the world. The researchers discovered that plant leaves with high photosynthetic efficiency have a shorter lifespan, but those with low efficiency have a longer lifespan. Subsequently, they uncovered the reason longer-lived leaves are thicker and require a muscular structure and cell wall characteristics that decrease photosynthesis efficiency to maintain their longevity. Long-living leaves possess thicker cell walls that impede the supply of nutrients vital for photosynthesis, particularly proteins, as they are delivered to the cell. In addition to that, thicker cell walls decrease the carbon dioxide available for chloroplasts, leading to a less productive photosynthesis process. The Methuselah worms, believed to be the world's longest living organism, were discovered underground in Cleveland, Findlay, England by British scientists in 2005. The Methuselah worms are bacteria that have been analyzed and identified as having a lifespan of 260 million years. Scientists discovered them in what was formerly a shallow area of water, and after the water completely evaporated, they were deposited along with salt. According to scientific belief, the organisms adapted to challenging underground saline conditions during their long evolution by consuming organic matter found in the saline. The metabolism of these organisms became highly sluggish, nearly zero. Methuselah worms represent one of the most exceptional instances. Generally, even organisms that maintain the lowest dynamic energy balance, photosynthetic efficiency, or metabolic rate still lose primitive energy with each chemical reaction in the cell. In other words, the cellular divisibility is gradually decreasing for all cells. The lifespan of any species relies on the maximum number of cell divisions it can undergo. The Hayflick limit refers to the limit for humans. When the primitive energy is exhausted, it means that the life of an individual has come to an end. The scientific community has been debating the lifespan and aging of organisms at length. Although none of the hypotheses have been verified to date, the telomere hypothesis, the free radical hypothesis, and the gene transcription error hypothesis are some of the better regarded ones. According to the telomere hypothesis, telomeres are distinct structures located at the ends of chromosomes, and their DNA comprises simple repetitive sequences. Telomeres get shorter progressively during cell division because DNA polymerase cannot fully replicate them. As cells divide, telomeres continue to shorten until their length reaches a specific threshold, at which point the cell enters the senescent stage. According to the free radical hypothesis, free radicals are atoms or functional groups that contain unpaired electrons and are usually formed in living organisms. They are diverse and plentiful, and act as highly reactive intermediates in transition states. Free radicals with high chemical activity can damage nucleic acids, proteins, and other macromolecules in living organisms by causing oxidative damage, DNA fragmentation, protein deactivation, and other effects. According to the gene transcription error hypothesis, as the body ages, the efficiency of intracellular DNA replication decreases, and large molecules such as nucleic acids, proteins, and enzymes are frequently synthesized with errors. These increasing errors can lead to a decline in cellular function, gradual aging, and death. The concept of primitive energy, which is proposed in this book, is not contradictory to the above three hypotheses. Telomeres could potentially act as carriers for storing the primitive energy. Reduced energy lock-in efficiency may cause free radical formation and gene transcription errors. The primitive energy consumption rate by cell division may not be constant. It decreases exponentially post-birth or development. This trend may continue until the organism dies, similar to radioactive materials decay. The genetically predetermined lifespan of an organism stays relatively constant unless there are significant changes in an individual's energy lock inefficiency caused by environmental changes. If the energy lock inefficiency varies significantly from that of its progenitors due to voluntary mutations, then the maximum lifespan of the organism will vary in accordance with its upper and lower limits. Replication and reproduction. Replication and reproduction are essential to the overall energy locking strategy, which, alongside the lifespan strategy, are the fundamental strategies for life continuation. Living beings carry on the best part of life from generation to generation in the form of energy seeds, which are essentially primitive energy. They do so by reproducing and multiplying. Merely transmitting the primitive energy and its operation mechanism is insufficient, thus the genes bear the responsibility. Genes record key codes related to the goal of primitive energy distribution order, and extraction method. Therefore, gene duplication is crucial for the continuation of life. Before cell division, gene replication of DNA double strands occurs, resulting in two identical double strands, if the process is natural, one of which corresponds to the original double strand. Gene replication mostly follows the pattern of semi-conserved replication. Each segment of the parent strand unwinds as a template, and by following the principle of complementary base pairing, relevant enzymes synthesize a segment of the child strand that is complementary to the parent strand. During the deconvolution process, the newly synthesized child strands are consistently elongated. Simultaneously, each child strand coils around its corresponding parent strand to create a double helix, resulting in a new DNA molecule. At the cellular level, rectification and error correction mechanisms ensure a high level of accuracy in DNA replication. Replication of DNA transpires at a defined location in the genome, known as the origin site. The DNA double helix comprises two opposing strands that run in opposite directions. Each DNA strand comprises 4 deoxyribonucleotides, A, T, C, and G. The base pairs of DNA involve matching the bases of both strands, which are A, T, and G, C. The replication of DNA involves 3 stages: initiation, elongation, and termination. During the first stage, helicases split the double-stranded DNA into 2 single strands. This allows the initiation enzyme to attach to the primer sequence of the single-stranded DNA and start replication. Right after that, DNA polymerase connects to the primer sequence and progresses, adding bases to form the new strand. As the 2 double helices are oriented in opposite directions, the polymerase moves in the backward direction on both strands. One of the strands, known as the forward strand, progresses continuously with the polymerase, with the newly replicated strand trailing behind. On the other strand, known as the lagging strand, the process involves binding, moving, and releasing in the opposite direction, while synthesizing a series of short DNA segments in a repeated cycle. Both polymerases on the strands work together during replication to maintain an almost uniform replication rate. Replication of DNA is not a miraculous event. It was not pioneered by our common ancestor. Replication is found everywhere in nature. All known matter is composed of atoms that have been replicated, and the same molecules in a material form are replicated from the earliest molecules formed. In nature, objects including organisms are repeatedly replicated, combined, and aggregated into the observed forms of existence. However, replication in organisms is highly self-organized, and the planning and control of each step of the replication process is impressive. Cellular self-replication is an essential mechanism by which organisms reproduce and transmit vital life information to their offspring. Through this operating mechanism, the primitive energy and its management system are informally stored in the DNA, taking an essential step in the continuation of life. The subsequent phase is cellular division. Cell division is triggered by the semi-permeable cell membrane, allowing small molecules to pass through freely while restricting the passage of large molecules. Accumulation of energy and information beyond a certain threshold in germ cells enables precise and periodic cellular division, rendering stable inheritance feasible. Cell division encompasses 3 well-known modes: mitosis, amitosis, and meiosis. While amitosis is utilized by prokaryotes, mitosis is the preferred mode of eukaryotes. Mitosis is a valuable type or stage of cell division that is characterized by no alteration in chromosome ploidy. Meiosis generates germ cells, or gametes, with half the required number of chromosomes for sexual reproduction. Meiosis represents a significant advancement in the technology of biological replication. Meiosis ensures relative genetic stability of species by preserving a constant number of chromosomes between parents and offspring. During each meiosis, non-homologous chromosomes are reassembled and some homologous chromosomes are exchanged. Meiosis facilitates genetic diversity in gametes and offers the essential technical support for organisms to mutate and increase their adaptability to the environment. In reality, biological communities do not always conform strictly to how biologists classify reproduction. Asexual reproduction is the evolutionary pathway that reflects individual genetics, whereas sexual reproduction is the pathway that reflects population genetics. In relatively simple multicellular organisms, these 2 pathways coexist. Species that reproduce asexually generally exhibit lower levels of offspring trait diversity, whereas those that reproduce sexually tend to have higher levels of offspring trait diversity. Some species employ a strategy of alternating, partial, or flexible use of both asexual and sexual reproduction, depending on environmental changes. For instance, consider Hydra. Hydroids typically reproduce through budding when the environment is favorable. In unfavorable conditions, hydroids produce papillate ovaries and spermathecae for sexual reproduction. After fertilization in sexual reproduction, the eggs develop into shelled embryos that enter a dormant stage until the environment becomes favorable. Later, the embryos develop into new individuals. In rare cases, hydroids can temporarily produce two reproductive glands in their outer body layer, ultimately resulting in the production of both eggs and sperm. Despite being hermaphroditic, partial chromosome exchange can also occur. Some animals exhibit both hermaphroditic and parthenogenetic reproduction during different periods of their lifetimes. For example, during spring and summer, Daphnia is typically observed as females only. They reproduce asexually by laying summer eggs that can directly develop into adults without fertilization. These adults are mainly females that can reproduce again asexually. A few smaller male individuals hatch from the summer eggs and initiate hermaphroditic reproduction during fall. The aphid is another instance that's parthenogenetic throughout most of its life and engages in bisexual reproduction only in late autumn. The eggs produced unite with sperm, remain inactive during the winter, and then develop into female aphids the subsequent year. Some species, like honeybees, exhibit both bisexual reproduction and parthenogenesis. They can produce eggs that develop into males without fertilization, while fertilized eggs develop into female workers or queens. Asexual reproduction's advantage is that it preserves all genes without loss and doesn't use as much time and resources as finding and selecting a mate. The mate selection process in sexual reproduction is energy-intensive, and the competition for mating rights can lead to injury or even death. Although the mate selection process in sexual reproduction can be strenuous, only half of the genes are transmitted to offspring. Species that opt for asexual or short-term asexual reproduction can allocate more resources to the reproduction of offspring. These organisms reproduce rapidly, with the number of offspring increasing drastically in a brief period. For instance, bacteria multiply every 20 minutes, which is a reproduction rate much higher than that of mammals. This breeding strategy based on population makes a lot of sense for less advanced animals. The lack of genetic diversity is a disadvantage in asexual reproduction, but it provides a numerical advantage for the offspring while reducing their risk in the survival environment, hence improving their chances of survival. As long as there is a small number of surviving offspring, the survival of the species is secure, except in cases of extreme and widespread risk. Once the environment returns to normal or migrates to an environment with sufficient energy sources, the surviving individuals start reproducing in large numbers and continue to do so. Moreover, this reproductive strategy offers the advantage of a well-developed military industry. If the energy supply is sufficient and the environment is highly conducive to reproduction, any individual employing asexual reproduction may transform into a warrior factory. Such individuals generate warriors on an enormous scale, besieging resources across all available energy loci and leaving no space for rivals. Due to the high degree of cellular differentiation, sophisticated animals are limited to a single reproductive strategy, namely sexual reproduction. Despite being time-consuming and taxing on resources, this reproductive process prioritizes the quality of offspring rather than its quantity, similar to the formation of an elite force. Sexual selection can be considered as a genetic contest for desirable genes. Competition for favorable traits, fitness, strength, and intelligence is essential for the victorious genes to propagate and extend over larger populations. In each generation, the selection process continues to choose the best players at a higher level of competition. The recombination of non-homologous chromosomes during sexual reproduction produces many genetically diverse players with different strengths, ensuring a highly competitive and entertaining selection process. Additionally, sexual reproduction optimally prepares a species for improved adaptability by increasing genetic diversity. The greater the genetic diversity of a species, the higher the degree of individual trait variation and diversity in survival sub-strategies. A species has a greater chance of survival if its population has higher genetic diversity, particularly during significant environmental changes or when facing an outnumbering attack from an asexually reproducing species. In terms of a species' probability of survival, the qualitative strategy of sexual reproduction is equally advantageous to the quantitative strategy of asexual reproduction. Interest and preference are also unique to each individual in sexually reproducing species due to the high degree of genetic individualization. Every individual has their unique need for and way of connecting. During courtship, the selection process is unlikely to be a limited event-based competition, and not all heterosexual individuals prefer every winner of each selection process. Instead, courtship selection is a comprehensive competition of interests and preferences. Behind each competition, there is a concealed rivalry at the level of the relationship. In humans, a unique term, love, is used to describe this competition at the relationship level. It is said that other animals have similar courtship behaviors. However, currently humans can't precisely describe them due to difficulty in observing and recognizing their language and behavior. Different languages enabling more complex communication and the pursuit of mutual interests and preferences are the core factors for successful pairing between males and females. Easing between leaps. Heredity and mutation are the two main threads of biological continuity and evolution. Mutation is a sudden heritable variation where a new gene replaces an existing gene at one or more loci. Mutations occur during cell replication and division when the DNA molecules on the genome undergo structural changes in the combination or order of base pairs, including site-specific mutations caused by changes in a single base, as well as the loss and repeated insertion of multiple bases. Variation is a prerequisite for evolution, and this is the unchallenged agreement of the scientific community. Despite its importance, Darwin's theory of natural selection has not yet been able to address the fundamental causes of variation and the mechanisms by which it occurs. There are two main theories of variation, gradualism and mutationism. Each has its views and endless debates. The most common objection to the doctrine of natural selection, according to American biologist Theodosius Dobzhansky, is that it does not explain the causes of genetic variation, but merely acknowledges its existence before natural selection comes into play. What makes it difficult to reconcile gradualism and mutationism? In the evolutionary process, genetic variation never intentionally ceases over a long period of time. There are abrupt new traits with large genetic differences between generations, and adaptive differentiation of traits with weak genetic differences between each leap. Mutation and gradual change can be likened to the 5 lines and musical notation of a score that together produce the beautiful and rhythmic music of life throughout the biological evolution history. Natural selection, on the other hand, is similar to how nature judges the music based on its preferences and ultimately removes what it does not like the most. Genetic mutations are frequently caused by errors that occur during gene replication when cells divide or from exposure to chemicals, radiation, or viruses, according to scientists. This is obvious, as sudden changes in the form and structure of energy in the environment may impair the accuracy of the replication process, as scientists have noted. This damage may even have fatal consequences, as explained by scientists. The concept of a replication error representing the human ego, without a solid foundation, is purely fictional and therefore perplexing. Organisms' life operating mechanism has been improving over billions of years and can precisely manage every stage of biochemical reactions, directing and executing every aspect of replication with a degree of accuracy much greater than the present-day man-made precision instruments. Considering how precision-driven heredity is, it becomes paradoxical to regard mutations as random and accidental replication errors. Variation is potentially the most illustrative quality of biological self-organization, indicating an active quest for change rather than a passive and unpredictable one. At this point, one might inquire, would proactive change neglect the impact of the environment? On the contrary, proactive variation is a strategic approach to respond to environmental change proactively. How can organisms discern what nature intends or favors when making decisions? How can organisms predict the environmental changes, as nature seems to know, before selecting an option? The laws of selection and environmental change are hidden somewhere in the universe, and nature has wrapped up these secrets in uncertainty. It may always remain a mystery to solve for every living species. What should be done? Through either wisdom or luck, the common ancestor of all living things developed two evolutionary strategies that were sufficient for their continuation. How is variation achieved? One potential strategy is that of genetic stability layering. DNA comprises two long helical strands that are twisted and overlapped into a supercoiled form. This structure's primary function may not be to facilitate storage in the nucleus, but to form a hierarchical inner and outer structure. Thus, this mechanism safeguards the core coding region's complete stability, while enabling the outer, non-coding region to monitor and document the dynamic changes in the environment. Some known species' genomes can only construct a small proportion of protein-coding exons from their core coding regions. Only 1.5% of the human genome, for instance, is reserved for protein-coding exons, while non-coding repetitive sequences make up more than 50% of the genome. In eukaryotes, DNA is organized into closely knit chromatin structures by binding with structural proteins. The double-stranded DNA attaches to a histone surface and forms a disc-like complex, called a nucleosome, by wrapping around two full turns. It then further spirals to form a highly helical, multilayered structure of fibers. The hierarchical structure protects the core coding region's absolute stability and implements the genetic variation's hierarchical mechanism in biological life. Therefore, genes should be classified into four groups based on their function and hierarchy: steady-state genes, substable genes, species genes, and environmental genes. Steady-state genes represent the central coding region of DNA fragments, located in the innermost layer of the DNA convolute and responsible for the fundamental mechanisms of life in organisms. These genes contain the essential genetic information necessary for synthesizing nucleic acids and proteins, controlling biochemical reactions, and primitive energy distribution activities, such as energy balance, intake, lock-in, and information processing, all of which are crucial for life continuity. Comparing multiple genomes shows that genomic differences between species are not significant, indicating that all steady-state genes may have been inherited from a common ancestor in the distant past and remained unchanged until the present time. Substable genes are potential elements of energy balance strategies and related mechanisms evident across various taxa, including kingdoms, phyla, classes, and orders. They record generic information accumulated over many generations, from a common ancestor to the present generation. This information concerns energy balance strategies that confer survival advantages, such as protein synthesis, biochemical reaction control, and energy allocation, among others. Genes specific to each species are found in the third layer of coiled DNA. This genetic material records information created by the recent common ancestors of families, genera, and species in evolution. The inherited genetic information from the first few thousand to the first few hundred generations is what has been preserved and passed down through the generations. The genetic information of species genes may mainly relate to sub-strategies of energy intake and lock-in. These relationships are expressed more in trait characteristics that are more specific when compared to those of other closely related species. Other DNA fragments that seem irreducible with individual characteristics, known as environmental genes, are attached to the outer layer of species genes. These segments are the centers for aggregating and processing various types of information received by an organism's sensory systems, that are equivalent to the primary centers for processing environmental changes. Environmental genes may not be transmitted directly to offspring, but can still transmit processed, fluctuating information to them. The primary processing center passes on the information to the corresponding segment in the species genes and screens, and edits it only if the energy stimulation in the environment is frequent or strong enough to reach the threshold to initiate gene editing. Once all the species gene encoding has finished, and the threshold for the initiation of editing by substable genes has been exceeded, the species genes transfer the task to the substable genes. Substable genes perform the same function with information as species genes. Completing the coding in one generation will cause the substable gene to rewrite the secondary life mechanism and to be passed to the next generation during cell division. The same applies from substable genes to steady-state genes, but Earth's environment may not have changed sufficiently to trigger a rewrite of the original life mechanism. The initial process of genetic information transfer and editing resembles a hierarchy of intelligence management. The receptor of each organism functions as an intelligencer, collecting information about environmental changes and transmitting it to environmental genes, which function as intelligence analysts. Environmental genes perform the primary processing of all information. Once processed, they report the summarized data and trend analysis to species genes, which serve as department heads. When the intelligence analysts report the same information more frequently, and the changing trend becomes substantially transparent, the department head makes a decision to adjust the strategy directly and implements it. If the adjusted strategy is still ineffective and the survival situation continues to worsen, the department head will report to the substable gene, which is the equivalent of the top intelligence center, and the substable gene will make a crucial decision to adjust the strategy. Genes adjust their response levels for each layer of information filtering, sorting, analysis, and transmission, from the most outer environmental genes to the substable genes, based on the severity of environmental changes. The genetic information related to environmental genes, species genes, and substable genes can be modified or deleted during the editing process. This information records the gradual changes in the cascade or stage that leads to the mutation trigger. Evolution is not solely a continuous and gradual process, but rather a combination of leap and gradual evolution, as determined by the macromolecular architecture of organisms. For instance, the first appearance of light-sensitive cells is marked by significant evolutionary leaps. Subsequently, a wide range of eyes and visual capabilities evolve asymptotically. Species evolution results from the combination of individual and population evolution. Sexual reproduction is not only important for increasing diversity through genetic reassortment, but also crucial because germ cells carry gene-related information from both sexes. Information from males may be predominantly correlated to environmental and species genes, while information from females might be predominantly correlated to steady-state and substable genes. In all mammals, premature eggs are present in the ovaries at birth, and the quantity of eggs does not increase after birth. In humans, eggs remain in the ovaries for several decades before ovarian function fails. Despite the adult cells having aged and died multiple times, the eggs remain safeguarded by the ovaries and secure from the various energetic impacts of the environment. Thus, it is reasonable to assume that the secret of heredity is associated with the egg. Substable and steady-state genes remain concealed within the egg, preserved by the ovary, the outer membrane, and the granulosa cell layer, sustaining the genetic stability of the core life mechanism in the mitochondrial DNA. Although eggs may carry some information related to species and environmental genes, these amounts are very small. However, the secret to variation may be most closely and significantly associated with the spermatozoon. This is because the spermatozoon is the primary carrier of environmental and species genes that document information on changes in the environment in which it exists. This information, recorded on environmental genes, which are most associated with individual survival hardship, can pass on information about the parent's environmental adaptations to the offspring. These species genes, which have the greatest correlation with population survival, can pass on the likelihood and direction of variation to the offspring. Sex differences resulting from sexual reproduction, a natural physiological mechanism for division of labor, are manifested in sexual traits. Additionally, sex differences in physical characteristics such as body size, height, muscle mass, endocrine system, and brain structure, including volume, cortical thickness, and neural connections, are also observed. The intangible traits of the two sexes are reflected in language, thinking, personality, and emotions. Sexual reproduction is a significant innovation and achievement for organisms in terms of adaptability and continuity from generation to generation. The common ancestor who first realized sexual reproduction inherited this evolutionarily significant mechanism after much debugging, making it a leap in genetic evolution. This mechanism splits the four core elements of sexual reproduction into two, endowing females with more sensuality and stability, and males with more rationality and pioneerism. However, individuals do not rigidly adhere to these elements based on their sex. Sexual reproduction allows for flexibility and regulation, enabling the structure and proportions of a small number of individuals to be appropriately adjusted based on specific circumstances and trial and error strategies. Biological sex differences play a crucial role in life by creating a reproductive division of labor and a balance between genetic continuity and mutation. In contrast, sociologists use the term sex differences to refer to a social division of labor that only considers the roles, status, and effectiveness of that division. Furthermore, sperm is believed to be the primary carrier of primitive energy required for offspring. The initial argument behind this claim is that during sperm development, certain spermatogonia can turn back time and transform back into stem cells, which is only possible through cells that carry primitive energy. The second argument emphasizes that solely the sperm with sufficient energy and appropriate movement can be the first to approach the egg's position and fuse with it, leading to the formation of a fertilized ovum, which can only be achieved by the sperm with maximum primitive energy. Throughout the history of biological evolution, numerous significant genetic advancements have been made, including macromolecular chains, cell membranes, cell division and differentiation, neural networks, sexual reproduction, symbiotic cooperation, brain and skeleton, among others. These evolutionary transformations have made organisms' energetic structures and strategies more intricate and resulted in a range of species. Major evolutionary leaps signify the start of substable gene editing. When species genes accumulate minimal changes and conditions are optimal, the triggering of substable gene editing results in trait differences that are taxonomically significant. Let us examine the evolution of photoreceptor cells. An ancient ancestor synthesized phytochromes billions of years ago. After many replications, they gradually assembled a screen behind the light-sensitive spots on the body surface. This screen continuously received incoming light, providing it with the initial awareness of light sources. After prolonged exposure to light in the environment, photoreceptor cells with phototransformation ability eventually emerged. This represents an evolutionary leap that occurred as a result of substable gene initiation editing. Worms and some mollusks transport cells that contain phytochromes to a tiny shallow cup on the body's surface, a setup that facilitates light direction detection. These cells then distribute the task, with each one responsible for sensing light in a unique direction. Hence, a small collection of light-sensitive cells on the same plane transforms into a shallow cup, which further transforms into a deep cup. The emergence of the eye lens represents another evolutionary leap in the history of the visual system. After the evolution of the simple primitive eye lens, species with this configuration temporarily have an advantage in terms of energy locus expansion and competition. They interact more frequently with the environment and record richer and more complete information on the environmental genes. Once a sustained positive feedback is formed, the evolutionary effort intensifies and continues to improve and gradualize in this direction. The mechanism that organisms use to avoid inbreeding can provide insight into the principle of genetic stratification in action. Sexually reproducing organisms appear to be born with this mechanism, and typically, animals avoid repeatedly reproducing between inbred parents within the same population over time. The same is true for plants, although this mechanism is less apparent in them. While insect, wind, or water-dispersed pollen does not completely prevent inbreeding, it can permit at least some pollen to reproduce at a distance. Over time, repetitive inbreeding can decrease genetic diversity, encourage the expression of undesirable recessive traits, and further deteriorate population fitness. Inbred individuals grow up in highly similar environments, while their gametes store almost identical genetic information from the environment. Overbreeding increases the probability of misinterpreting environmental changes, disrupts the activation mechanisms of environmental genes, and may even lead to incorrect initiation of genetic modifications in the species. As a result, repeated breeding can produce offspring with recessive and harmful traits, which have been observed in both plants and animals. Mistakes in evaluating the degree of environmental variability can result in negative consequences, hindering the population's ability to adapt to the present environment. Is the excitement of environmental change information in a species' genes the only condition for initiating editing? This might not be precisely accurate. Despite the evolution of all species, complete adaptation to the environment remains impossible. The system of an organism cannot perform complex mental processes such as measurement, analysis, experimentation, calculation, and logical reasoning about its surroundings. Moreover, it cannot collaborate or overlap in pursuit of a high scientific capability such as human knowledge. Furthermore, many areas are beyond human reach, and our bodies cannot carry out independent evolution outside of nature, based on predictions of the environment using our brains. The second evolutionary strategy involves preemptive directionless mutations that are the result of a voluntary mutation of a subset of genes. Voluntary mutation contains two meanings. First, it refers to the random fine-tuning of a species' genes during reproduction, which is triggered by the adventurous curiosity of unstable genes to achieve proactive, intentional, preemptive, and directionless mutation. Second, voluntary mutation aims to increase the diversity of a species' genomes to optimize survival through trial and error. In other words, the goal is to actively provide nature with multiple options to test and select. The significance of voluntary mutation is to decrease the chance that the genes of a species within a population suffer extinction in extreme conditions. In case of an environmentally abrupt change or an intense attack by another species, the diversity of traits at least ensures that individuals carrying a particular gene that is better adapted to that change, attack, have a higher chance of survival. This will allow the population to have a higher probability of surviving in the new environment as it spreads. Mimicry, which we observe in organisms, is an incredible survival strategy. It may not always be a result of mutation in response to environmental change, but a result of self-initiated mutations in populations migrating to more favorable environments to ensure survival. Evolution can be considered a purposeful mutation process resulting from a trial and error approach. The trial and error process can be randomized based on the normal distribution curve, which represents significant variation values on both sides of the graph. When the population is adequately large, even slight variations and their corresponding traits can become manifested on the graph. However, these traits should not be discarded as anomalies, since in nature, which is full of unpredictability, any trait may survive or die off. Early human ancestors who were apes may have been regarded as freaks by other apes when they began to walk on two legs with a toddling gait. However, a subset of these abnormal descendants eventually became the predominant species on Earth millions of years after the dinosaurs of the Jurassic period, similar to their rise to dominance. Therefore, we should not hastily categorize certain individuals' unique qualities as illnesses, nor should we regard such individuals as defenseless, such as those with autism, pseudo-hypertrophic dystrophy, or other congenital or genetic traits. Each individual has a unique set of traits, much like fingerprints, with some traits being more noticeable than others. Perhaps in the future, under extreme circumstances, individuals with a specific advantageous trait could contribute to the continuation of the human race. Academics fixated on evolutionary theory tend to generate hypotheses after the fact about biological attributes within a given environment, therefore unconstrained by reality. Whether any variation is advantageous, detrimental, or neutral is immaterial. There are no predetermined advantages or disadvantages to any options before natural selection occurs. Animal eyes take on different forms among species, which include the compound eyes of arthropods, the translucent ones of vertebrates, the dark-chambered eyes of nautiluses, the vesicular eyes of snails, among others. It is difficult to determine which visual organ is better suited to the environment, or whether the corresponding genes are more harmful or favorable. The only certainty is that the least adaptable individuals have likely been eliminated over time. Although there is a common belief that natural selection favors the best, it may, in fact, aim to eliminate the worst. Not all genes, traits, or survival strategies in living species on Earth are optimal, but also not the worst overall. Nature does not strive for perfection, but only selects through elimination, probably contributing to the rich diversity of living species on Earth.